we did a study of Sacituzumab govitecan, also known as IMU-132. It is an antibody-drug conjugate, so the antibody recognizes an antigen called TROP2. TROP2 is expressed on epithelial cells and is overexpressed in a number of different tumor types, including urothelial carcinoma. The antibody is linked to SN38, which is the most active metabolite of the chemotherapy irinotecan. And with that antibody-drug conjugate, it's given systemically and is preferentially taken to areas of excess TROP2 expression. So the mechanism of action is a cytotoxic that is preferentially delivered near tumors. So overall, the study was what many people call a basket study initially. So there was an initial phase 1 dose escalation component where a dose of 10 milligrams per kilograms day 1 and day 8 of a 21-day cycle was determined. And then there were a number of different tumor types that looked interesting. So we saw 6 patients in the initial dose escalation phase of the study that interestingly did much better than we expected. So this is in the setting of not having a lot of options for advanced urothelial carcinoma, and that led to an expansion cohort or a phase 2 cohort of this drug. So we're presenting essentially a phase 2 study of 45 patients with refractory advanced urothelial carcinoma who received IMU-132 at the recommended phase 2 dose. What we found is a number of things. I'll separate efficacy and toxicity. So efficacy I'll start with, which looked impressive. So this is a non-randomized study, but based on what we expect for patients that have received the standard therapies before. So this was started in an era where there was really just chemotherapy. And then later, immune checkpoint inhibitors came into the market. So in the tail end of the study, there were patients that also had received those. A median of 2 lines of prior therapy, but up to 6 prior lines of therapy. There was no limit, provided the inclusion criteria were met in terms of performance status and organ function. And we saw an impressive response rate. So kind of historically speaking, we'd expect that if I were to give someone an available chemotherapy agent, we'd expect doing a study, we'd expect approximately a 10% response rate. What we saw overall in these 45 patients was an approximate 30% response rate in this heavily pretreated patient population. And we looked at the subsets. So these are not big subsets, but the vast majority, about 3/4, had visceral metastasis, including a third with liver metastasis. Those that had or hadn't received prior immunotherapy, the response rates were similar in all of the different subgroups. So it looks like it works despite sites of disease as well as prior therapy. Adverse effects. So if we concentrate first on the more serious, so grade 3 and 4 adverse events, there was no non-laboratory adverse event that was in the double digits, meaning above 10%. Diarrhea was about 9%, but lower than we'd expect with the free drug irinotecan. That likely has to do with the drug delivery mechanism, that it's not going through the liver. So that's likely, but we didn't see as much diarrhea as we might with the free drug irinotecan. We saw essentially no significant neuropathy or anything else. What the dose-limiting toxicity of the drug that was discovered in phase 1 and also confirmed in phase 2 is myelosuppression. So there is a reasonable amount of grade 3 and grade 4 myelosuppression. Did not result in a lot of neutropenic fevers, fortunately, but myelosuppression is the dose-limiting toxicity of the drug, reasonably easily managed with either dose reduction or the addition of growth factors. So this was, you can call this a proof of concept study. Only 45 patients is maybe it was patient selection why it looks so good. So what has been launched now is called the TROPHY U01 study, which is a more definitive phase 2 study with the primary cohort that's going to enroll 100 evaluable patients. There's a poster also on this today, looking at the specifically patients that have received both platinum chemotherapy as well as a PD-1 or PD-L1 antibody. There's no limit to additional therapies, but at least having those two types of drugs separately or together, and we're looking to really confirm the response rate as well as tolerability of the drug. There's a separate cohort of non-cisplatin eligible patients that's more exploratory, that is just after immunotherapy. But really what we're considering the potentially pivotal cohort is this cohort of approximately 100 patients that could, at least in theory, lead to registration. So the initial basket study looked at a number of different cancers, and many of them looked interesting. And so in a refractory patient population, there were responses across the board. The single biggest data set was in triple-negative breast cancer, where there's now hundreds of patients that have been treated, and that has been submitted to the FDA. And we think that there's a reasonable chance that that may lead to approval. Urothelial, we're hoping, as a GU oncologist, I'm hoping that will be the second label. It's also being explored in other cancers. So, like I said, a number of different solid tumors did respond to the drug. There is a new study that's being launched that is selecting for TROP2 expression and then enrolling those studies that will include lung, GI cancers, and various other cancers.